In the previous video, we discussed about glycolysis and how that process was able to generate two ADPs for us. And just to get a little bit of overview of what happened in the previous video, what the first step was that we had a 6-carbon chain glucose molecule, and it went through a process of 9 steps, and along that process we had an NAD plus reduced to NADH. Remember, reducing means gaining an electron, and actually there were 2 of these, so we had two, and we had 2 ADPs being reduced, 2 ADPs changed into 2 ATPs by gaining a phosphate group. So phosphate, 2 of them here, and we get 2 ATP. So the net, and at the end product we had a 3-carbon chain molecule, which we call pyruvates. Each of these which we call pyruvates. So at the end of glycolysis we had 2 ATPs and 2 NADH. Now in this video we're going to discuss about the process which occurs in the mitochondria, and that process is called the citric acid cycle. And we're going to discuss that why is it called citric and why is it called cycle, and another name for this process is called the Krebs cycle, named after the scientist Krebs, who told us much of its process, the Krebs cycle. So now, before going into the citric acid cycle, this pyruvate right over here has to be modified. It has to go under some kind of processes in order to be able to enter into the citric acid cycle. And remember, this process, the citric acid cycle, is happening in the mitochondria, whereas glycolysis was happening in the cytoplasm. So before going into the citric acid cycle, the pyruvate, so I'm just picking up one of the molecules, goes under a process of 3 processes happens so that it could be ready to enter into the citric acid cycle. And one of the processes is that as it's going through a process, it releases carbon dioxide. So one of the carbons, right, we can say this one, is released. It leaves the 3-carbon molecule as carbon dioxide, and actually this is the first part of respiration where we witness carbon dioxide. As we have discussed in earlier videos, that respiration is the process of breaking down of glucose into carbon dioxide, water, and energy. So this is the first point where we see carbon dioxide. The second point is that an NAD plus reduces into NADH. So you can just add another NADH here. So we have another NADH. And the third process is a coenzyme A called CoA, it's derived from vitamin B actually, and it joins this 2-carbon chain molecule because one of the carbon has left as in the form of carbon dioxide. It joins the 2-carbon chain molecule, and then we get this molecule which is called acetyl CoA. We call it acid because it has a 2-carbon molecule, 2 carbons. So right here. So now this pyruvate right here, for this we are ready to enter into the citric acid cycle. Now, first of all, when the acetyl CoA enters into the citric acid cycle, which is in the membranes of mitochondria, so it joins a 4-carbon chain molecule. So 1, 2, 3, and 4. It joins a 4-carbon chain molecule called oxaloacetate, and it joins together and forms a 6-carbon chain molecule, so 2 and 3. 2 and 4. 1, 2, 3, 4, 5, 6, called citric. Now you might be wondering where the CoA part goes. So as it enters into the citric acid cycle, the coenzyme A, it splits, it breaks off from the 2-carbon chain molecule and is recycled and reused again. Its main function was just to get the carbon molecule in the citric acid cycle. Now we have a 4-carbon chain molecule called citrate, so the word citric acid comes from here. As it goes on to the next step, we witness that an NAD plus is reduced to an NADH, and carbon dioxide is released from the molecule, and we get a 5-carbon chain molecule, 2, 3, 4, and 5, which is actually called, so you don't have to remember these are just, if you like to remember these names. So basically there's a 5-carbon chain molecule. It's called alpha ketoglucerate. Next, in the next process, we go from a 5-carbon chain molecule into a 4-carbon chain molecule, and in this process another NAD plus is reduced to NADH plus a proton, so you just have to mention the proton too. By proton I just simply mean a hydrogen atom with just one proton in it, so it's basically a proton. And carbon dioxide is also released, and in this process we are able to generate one ATP from an ADP. So this here is an ADP and we generate an ATP. Next, in the next process, we go from a 4-carbon chain molecule. So now we have a 4-carbon chain molecule, and this carbon chain molecule is actually called succinate, if I'm not pronouncing it wrongly. Next, from a 4-carbon chain molecule, we go to a series of processes in which an FAD, a new word which we learn here, an FAD is reduced to FADH2. It actually stands for flavin adenine dinucleotide. And you might be wondering what kind of molecule is that? It's actually an energy carrying molecule, and which has a lot of energy stored in it, and this FAD along with these NAD pluses are combined together in the electron transport chain, which is the next process where we are able to generate more ATP. So just for the intro of this molecule, so after the succinate, after series of the structure of the molecule keeps changing as it goes to different processes which occur by the help of enzymes, and we get another, it's still 4-carbon, but the structure is different. So now we get a molecule which is called malate. From malate, as we go on to the next step, another NAD plus is reduced to NADH. So we actually had 3 times, one here, and another one here. And this 4-carbon chain molecule goes back into its original shape, which is right over here. It changes its shape and becomes an oxaloacetate, again forming this shape right here. So now you might get an idea of why we call it a cycle, because we start off with an oxaloacetate, which is already present in the mitochondria, and we go through all this process and we end up back again to oxaloacetate. So there you go. This is the citric acid cycle. Quite easy and simple, just to have to know where these NAD pluses comes along. So just to get a little bit of overview, from glycolysis, we were able to generate 2 ATP and 2 NADHs, and from this process to preparing the pyruvate to go into the Krebs cycle, we generated 1 NADH. So this is only for one pyruvate. So if you're making 2 pyruvates, so we will reduce about 2 NADHs. So we now have 2 NADHs. Now, from the Krebs cycle, we were able to make 3, one over here, and another one over here. We were able to make 3 NADH, sorry. And NADH only for one pyruvate. So if we're talking about 2, so we're basically talking about 6 NADHs. Remember, 3 for one pyruvate, 3 for another pyruvate. So at the end of this glycolysis and citric acid cycle, we have about 2, 4, and 6, and 10 NADHs. Next, if you talk about in terms of ATPs, or the currency of energy, the energy-rich molecule from which we get energy, we generated only one. Here it is. We generated only one ATP. So if you want me to do another color, here it is. We generated only one ATP. So we generated one ATP, right? So this one is for only one pyruvate molecule, which I mean like only one for the acetyl CoA. We're talking about 2 pyruvates, which actually we break down glucose into 2. So we generated 2 molecules of ATP. So at the end, we generated 2 ATP molecules from the Krebs cycle, 2 from glycolysis, 2 from Krebs cycle, and at the end we have 4 ATP molecules. And so we made a new molecule right here, and yes, this one's right here, the FADH2. And remember, this is an energy carrying molecule which has a lot of energy stored in it, which we're going to get that energy when we go into the electron transport chain. And so we generated only one. So if you're talking about 2 pyruvates, so we generated 2 FADH2. So at the end, we had, just to make it a little bit cleaner, we had, here it is, 10 NADHs, 4 ATPs, and 2 FADH2s. So this is our product which we were able to receive from the whole process: 10 NADHs, 4 ATPs, and 2 FADH2s. And in the next process, so I hope you guys found that useful. And in the next video, we are going to discuss about the electron transport chain, from which we will be able to generate a lot amount of energy. Probably I think it's 38. But so hope you find it useful, and see you in the next video.